Can you see my screen?
Yes, sir.
Yes. Okay. In the last class, we discussed about prostate cancer stem cells, and then we learned about some of these types of cells that are there. The cells such as the luminal cells, which are the actual functional ones that produce the prostatic fluid into the lumen. We spoke about basal cells. This is the layer of basal cells that is stuck to the basal lamina. And we also discussed about neuroendocrine cells that kind of integrate or to tell the prostate gland when to produce more, less, and so on. We also discussed about the intermediate cells, transit amplifying cells. And then we spoke about the cell surface markers of the stem cells, right? And in that case, we discussed about the proliferative potential. And the highest proliferative potential is for the cells that were having CD44 plus, integrin alpha beta 1 high, and CD133 plus. And we also discussed about what is meant by proliferative capacity and proliferation rate. So proliferation rate is high for transit amplifying cells. Proliferative capacity is high for the stem cells, and the differentiated cells do not have either of them. So in tumors, the cells, if you're considering a cancer stem cell, you can see that the cancer stem cell falls in this line, which has high proliferative capacity. That means they can divide continuously for theoretically innumerable number of times. The rate of proliferation, however, is very less. They will divide only when there is a need, kind of thing. The tumor progenitors, which fall or which are analogous in the sense with that of transit amplifying cells, they have high proliferation rate. They divide very rapidly, but they have very less proliferative capacity. The difference between these two I have discussed yesterday. Proliferative capacity is the number of times it can divide. Proliferation rate is number of times it can divide per unit time. So this is basically the proliferative capacity is indicative of stemness. So these are the tumor progenitors, like the transit amplifying cells, they divide much more rapidly. And then they eventually will differentiate into some things, they will live for a while, and they will die. This is the traditional or general cancer stem cell model. We also discussed about androgen ablation, how to cut it out, because the luminal cells are highly dependent on androgens for survival. So if we remove the supply of androgen or testosterone, somehow those cells will die. But somehow the malignant cells might have acquired mutations, because of which they are no longer dependent on the androgen. And then they will grow whether androgen is present or not. So we discussed also about testosterone and how androgens, they're taken in testosterone, for example, acted upon by 5-alpha reductase, dihydrotestosterone, then it binds to androgen receptor, dimerization and phosphorylation, and then it goes into the nucleus where it will act as a transcriptional factor or activator by binding to androgen response element. And then it will allow the expression of several genes involved in growth and survival. We also discussed about some of the methods that are there to control prostate cancer. So we discussed that there could be, every cancer, there is a high degree of mutations that will happen, and those mutations could cause the androgen receptor. Sometimes you may have amplified androgen receptor overexpression, because of which there will be more and more of activation of the target genes. That is the hypersensitive one. Promiscuous is if there is any mutation, because of which this affinity has changed. Androgen receptor is supposed to bind to something, a molecule like testosterone. But now, because of the mutation or change in the active site, they are binding to other molecules. And if that other molecules like these are present, it might get dimerized, activated, and then transcriptional activation of target genes. There are also other ways where, because of mutations, they might be influenced by other molecules or other signaling pathways, and because of which also the hyperactivity and then cell division and survival may happen. So there are some mutations like this. T is substituted by A. Can you recall if it is transition or transversion? Okay, it is at the level of protein. Okay, this is not at the level of DNA. Usually, this is the amino acid threonine replaced with adenine. Sorry, not adenine, alanine. Okay, this will be leucine replaced histidine. That's right. And there are also some cases where double mutants may also happen. So what was actually should be binding to testosterone is the mutations because of which they're changed to some other molecule like this. And in another one, it is now binding to corticosteroids. Okay? Yes. So this is the end of prostate cancer stem cells. All you need to remember in this particular chapter is the signaling pathway here. And prior to that, we should know about the cell surface markers of these types of cells that are present, the presence of stem cells where they are, and the functions of prostate gland in general. And then we should, of course, know the ways to how to find out the cell surface markers or isolate the cancer stem cells. That's a general topic that we have discussed in the previous chapter. And then we have to know about the probable cell surface markers of prostate cancer stem cells. That would be CD44 plus, alpha 2 beta 1 high, and CD133 plus. And you should know about proliferative capacity and proliferation rate as the terms, but otherwise we discussed the concept many times before. Androgen ablation, dependency on androgen, and I want you to know the signaling pathway. This is simple. All you need to remember is alpha reductase, 5-alpha reductase, dihydrotestosterone, and then what happens to androgen receptor. And then the kinds of mutations and how the cell becomes androgen independent. That's the important point of it. It becomes androgen independent. Okay. We'll move on to the next another lesson, and I will give you an introduction, and I will stop here today. So we're going to talk about melanoma cancer stem cells. Melanoma is a cancer of the skin, and we will talk about the biology. And the important part in this is the signaling pathways, not signaling, hedgehog and Wnt signaling. So this chapter might take 2 or 3 classes. So the rest of the things, there will be a lot of what you call redundancy with what we have already learned. Okay? The first thing, as I was telling you some time ago, if we are feeling sick, and this I used to use in every class I discuss, because we are all influenced somehow, especially when we have no other work, then we start Googling and all the other stuff. We say during the COVID thing, if somebody sneezed, we would suspect that that person has COVID maybe, or somebody has cold. But if it happens to us, maybe we might think it's a general cold or allergic cold. That happened to me. I did that. So it's a general tendency during COVID. That's how we used to be suspicious about other people, because it's a pandemic. We are likely to get from others, and it is transferring very easily. There will be some in some point of time we might have headache or some things, and then we might get really scared, look up on the internet, and then we will come to a conclusion, brain cancer or whatever, and then we'll have a really serious trouble, because we get sick. The mind is unhappy and the body gets sick. And this is a nice one too. Googling your symptoms when you don't feel well is the most efficient way to convince yourself you're dying, right? And I also had a nice picture somewhere which says in front of the doctors, the near the board outside the doctor's office, it's written, please keep all your Google knowledge or Wikipedia knowledge outside and enter the room, enter the doctor's office. That's a nice way also. Anyway, so melanoma is abnormal growth of melanocytes, and it can affect several other tissues that are like skin. Skin is the most affected one, of course. Eyes, meninges, and digestive tract. So there are about 77% of deaths of the skin cancers. There are some of them are really aggressive. One of the main reasons is supposedly ultraviolet radiation. What we should also understand is these are statistics that would usually come from the Western world or developed countries, right? The underdeveloped countries do not have those many resources or focus as much as the developed countries have. And we should also see, as we will see in the geography, how it could affect as well. There are also genetic and familial genetic predisposition. We don't know it yet. And most are sporadic. So look at this first. What you would notice is this is supposedly somewhere here. I think the equator passes by, right? Somewhere here, I think so. And the skin color of these people is they are highly pigmented, dark, because we have so much of melanin. And the most of the Western world has come from here. This Europe, from Europe, they are Caucasian. Caucasians have very light or a very less pigmentation, melanin production. Why do they have less pigmentation? How did they get selected? People living in these conditions would actually get very little sunlight during the winter, right? In the winter, this daytime could be as little as 4 hours. And as you go to the North and South Pole, at one point or the other, during the winters, respective winters, they have very little sunlight. That means the person in the old ancient times was not receiving vitamin D. Sunlight is important for producing vitamin D, but it's also through sunlight you can have UV light as well. So people surviving in these areas should have, in the ancient times, of course, should have had, if they have lighter skin, then they might absorb more sunlight, isn't it? And then they will probably be able to produce sufficient vitamin D. So that is the logic of thinking that why the Caucasians have very light skin tones. And those living in the equator regions where the sunlight is plenty, harsh and hard, then they need to be protected, and melanin is the molecule that protects the skin and from these harmful rays, because there are also UV rays. They're getting more than required amount of exposure. That's why we have a pattern. But over the course of time, these Europeans have moved to several continents. Say into South Africa, they came into India also, they are in Australia, and so on. And if you see these people, they have the Caucasians have evolved their skin tone for the conditions they are living in, but now they have moved to different locations, right? And this is my understanding or my analysis, perception. It doesn't have to be correct. So I think these are the people who are likely or more, because they have very light skin tone, they might be more prone to skin cancers. If you see them, if they just walk sometimes, when I was in Denmark, I used to see many people complaining also. They just walk in the summer, a little bit out exposed, their skin becomes very red, a lot of sunburns. Okay, now we have chemicals or some kinds of creams to protect their skin, but otherwise, I think skin cancer probably is more associated with the Caucasians all around the globe. It's just my perception. Okay, so how does or what does it happen? They call it as ABCD: A, B, C, and D. Asymmetric, that is, we may get many things in. We also get sometimes some colorations on our skin, and the moles can be there. They are usually circular or some kind of symmetry is there. Whereas those of melanoma, they are usually symmetric like this. Second thing is the borders are irregular. In the moles, you have a smooth borders, whereas in here, you see that these are irregular borders like this. Third one is coloration. A mole in our normal one on our body will have almost uniform coloration, whereas in these, in melanoma, you might find different types of colorations. And a mole is usually smaller. When a new one develops, for example, they are less than quarter inch or say 1 centimeter or less than a centimeter, half a centimeter. Whereas this one, these are usually larger than that, or they grow into that. And the next part is evolution. That means it grows in size, right? One mole like this, and over a period of time, it keeps on growing. I want you to look at this picture and tell me what is odd about it. Is it a genuine picture or not a genuine picture? Anybody, any of you, please?
Sir, the last one or
All these pictures, these three, what do you see in them?
sir, by observing the texture and color, it looks like editing, I guess.
Yes, this is fabrication. This is the original picture. Okay, somebody wants to show evolution. Otherwise, everything is same, right? You see the lip marks, the everything, the pattern, everything is absolutely same. In science, there are also, as I was talking about integrity, there are many people who fabricate their data, the results. Science is not easy. Science is difficult. And many people take short routes to cheat the system somehow and then publish papers. This is probably not from a scientific journal, but people use the skills with computers using many different types of softwares, and this person has erased it here. Okay, from this part was erased of this picture, isn't it? So we should be very careful about what we believe and what we not believe. Science is a celebration of doubts. That means if some data is produced, some fact is told to you, or something is given to you as piece of new information, you don't approach it to accept it. You approach it to reject it. That means you're going with a a doubt whether it is not right, let me check it. Let me prove that it is wrong, is what the science approach is. There is also pseudoscience. They will say there, I think if I'm eating cashew nut, I will get better. And then we eat cashew nut, we get better. We don't know if it is because of cashew nut or not, but we will decide that cashew nut is the medicine for this particular disease. I'm just making up an example. So science is you have to approach everything with a sense of doubt. Okay. So this one is the idea is in this particular instance is to say that the melanoma evolves. It doesn't remain the same. It evolves much more rapidly in the sense it changes shape, texture, and all these things. It grows faster. But this picture also illustrates the nature of how some people may find shortcuts to publication success and so on. I think I discussed about staph cells, right? You have to remember these things so that we will never do these in our life. There is something called PubPeer also. You can go there, people commenting on finding faults with papers like image fabrication and data fabrication and so on. Anyway, let's get to it. So these are some of the pictures which illustrate these things. This is the normal ones, I guess. This side is the normal ones. Usually they are this way, whereas the melanoma ones are here. I think so. So if you are also into science and we have a course you have read in your first semester or second semester, biology for engineers, here in 2017 or so, yes, a paper was published in which they developed a program, computer program with neural networks, and that can identify if a picture of the skin cancer or a picture of a discoloration is sent, the computer can analyze whether it is a skin cancer or not, and it can also level. It has performed very well in comparison to the human doctors. Somebody is telling by experience. That's a good thing. So melanoma is happens in this way. So we have the skins, the epidermis, and the dermis. Outer layer can be the keratin layer, and then what we see is in this surface, but actually inside it keeps on growing inwards as well, and eventually it will reach the bloodstream, and then it will go into metastasis. So it may originate somewhere here, but then it will keep on going in, and then to the bloodstream stage 4, and from here it will move to the rest of the body, and establish cancers in other tissues as well. So as I said, this can also happen in eyes, on meninges, and so many other places too. So when it happens in the eye, it's a tumor-like thing can grow in the eye, and that will cause blindness, and there is only one way is to get rid of the eye in that case. We know about Michael Jackson, right?
Yes, sir.
Yes. How did he become fair? He's a black kid, right? He was like this, and how did he become like this?
He undergone a plastic surgery, I guess.
No, that's not true. I think so. He has undergone plastic surgery for several things like nose and things. He had his own desires to look something. Do you think if he purposefully changed from this to this, is it right or wrong?
No idea, sir.
No, you should. It's just an opinion thing. It's not
So it's wrong, sir.
Why?
So it's his nature, sir. Changing his nature for it's wrong, sir.
No, we all do that all the time.
So, but
We are-
not to this extent, no, sir.
No, know. How do you decide?
So maybe he faced some racism, like.
Assume it is not racism. Even if he faced racism, should he do or not? Okay, sorry for asking this question. Actually, it is not our business. It's his business, right? He's changing himself and he has the things to do because we all do our own ways. We groom ourselves, we clean our face out of all the oils, whatever our natural is. We try to look different too. Only thing we don't take to the extent like we said. But then what he does is his business. But the point is that he actually had a disease called vitiligo. He had, when he was transforming, you see some spots here. If you can, I hope you can see those. You can see some spotting here.
Yes, sir.
And this is his son who apparently has that. I don't know how true it is, but yes. So, and in his death report too, he had this disease of vitiligo. That is a discoloration of the skin. So this is a nice representation. A girl born like this, over some period of time, has become like this, full fair. Okay, that is, she lost the melanocytes, the pigment-bearing cells, pigment-producing cells are lost, and so her skin now is without pigments. That is a progressive kind of autoimmune disease where the immune system makes antibodies against the melanocytes, and because of which melanocytes are gradually killed away. And then what we are left with is cells without any pigment, skin without pigmented cells. Right? It is progressive and it keeps on happening until the body becomes completely pale. So, have you ever gotten hurt? When we get hurt in our knees, if we played outside and we lost a bit of skin like this, for example, and then we see growth happening. If there should be a big enough injury, I experienced this. And then you will find some small spots, right? After an injury, or if vitiligo is treated with some compounds or chemicals, then the pigmentation starts to reappear, and it's not happening from one place, it's happening from small tiny spots like this. If we look closely, this is how it is. It is happening around a hair, right? Here is the skin hair, and around that this pigmentation is happening. These are the spots. That means somewhere melanocytes are being produced, and they are traveling outwards like this. Okay. So this is a hair follicle. We have the hair, skin hair, and there are inner root, outer root, and so many other things, progenitor cells for the hair and all. And there is the sebaceous gland, which produces sebum, and we have epidermis and the basal layer and dermis and so on. We also have bulge, a small bulge here, this region. That is where the stem cells are. For producing all of the hair is also coming from these cells are supplementing, and also the skin cells as well. And that is where melanocytes also come from. So melanocytes are something like the migratory cells. Their skin is kind of very dramatic, right? Somebody becomes very fat, all of a sudden they'll find stretch marks. If they shrink down, the skin again shrinks. It is also a kind of dynamic one. One thing is, skin is always being lost to the outside. The outer cells are getting the keratin layer, and then the epidermis, and we have the dermis. These cells are growing more and more, and these cells are dying and falling off. That's a very common procedure. It is happening all the time, and it is happening more than ever because we use a lot of soap. No other animal does it as much as we do, right? And we are continuously, almost forcefully removing some things. We rub and all to keep ourselves clean. Of course, we are. It's like this: somebody, if we don't bathe properly every day, we will have a lot of infections immediately. When have we seen a dog do that? Not the pet dogs, but the street dogs. We don't see them all doing these things. Their skins are hard and many things. So what I'm trying to say is the melanocytes or the melanoblasts, they keep on producing, and skin is a dynamic tissue of the body with second most, I guess it is the second most regenerative, with the regenerative potential within our body. The first one being liver. If you remember, liver is um, one of the organs in the body which has most regenerative potential. In the first introductory lesson, we learned about that. I think I will give the second one is probably the skin. Okay. So it is thought, well, this is the developmental biology. We don't have to really go through it. The neural tube, and the neural tube, some of the cells are produced, and those are the precursors for the melanoblast, pre-migratory melanoblast, and then they travel out and into the skin, and then they settle into the bulge as melanocyte stem cells, and then they keep on producing more and more cells, so that will form the skin. Okay, I'll take this slide and then I will stop. So what we know of is we have a melanocyte stem cell that is present in the bulge region near the hair follicles. That gives rise to some cells that are something like the transit amplifying cells, which will again divide. And we have two different types of cells, the melanocytes, and those cells that have other functions to move around. So the melanocytes are the ones that produce the melanin that is important as a UV protectant. It may have other functions such as metal chelation, endocrine functions, and immune functions as well. In the last lesson also, we tried to understand where does the cancer stem cell arise from. We don't know for sure where the mutation would have occurred. Does it occur in the melanocyte stem cell, or does it occur in this cell stage, that is a transit amplifying or intermediate stage? We don't know that. So there is a possibility that some of the cells around this stage might have become mutant, and that is where the melanoma starts. And then they may move into the bloodstream, move into different locations, and it becomes metastatic. Okay, so I'll stop here. If you have any questions, we can discuss on it. And in the next class, we will talk about it a little more. And then the most important things will be about learning the cell signaling pathways. Okay?
Yes, sir.
Yes. I'll stop the recording.